Welcome, my dear student. Today I shall discuss about DNA. You have some idea about DNA. DNA is the genetic material of living beings. There are some viruses where the genetic material is RNA. But our discussion about DNA. But what one question arises that where the DNA is found inside the cell. Okay. So DNA is located within the nucleus. This is the cell structure. Within the one of the cell structure, cell organ is nucleus, and DNA found in cell nucleus. Nucleus contain chromosome, and chromosome within the chromosome DNA is present. One another question arises that there is any other organelle where the DNA is present, or where the where organelle like nucleus having own DNA? Answer is yes. That is chloroplast having its own DNA, mitochondria having its own DNA. I will discuss it later. But at first, I discuss structure of DNA. Before I start the structure of DNA, there are some historical perspective that not a single day nowadays we have some idea about DNA's double helix, DNA's right-handed helix, DNA is made up of nucleotide, DNA having phosphodiester bands, that is. But where it is short? Who first proposed that? Who first isolated the DNA, named DNA, or what is the concept of DNA? At first, 1869, Miescher first identified nuclein inside the human white blood cell. Nuclein means from the nucleus, but he did not discover it or did not discover its structure. Then in 1999, Levin discovered the order of three major components of single nucleotide: phosphate, sugar, and base. This is the main events in DNA discovery. That phosphate, sugar, and base are the major three components of DNA. I discuss later for this, but this is the nucleotide concept, propose the polynucleotide concept. After that, after that, there are one scientist, Chargaff. Chargaff proposed his rule, Chargaff rule. He told the total amount of purine, one of the bases that is having two ring structure, and pyrimidine, one of the other bases having one ring structure. That is, the total amount of pyrimidine and purine are nearly equal. Pyrimidine and purine are nearly equal. So that A is equal to T, G is equal to C. So A plus G total purine is equal to T plus C total pyrimidine or pyrimidine. At last, 1953, Watson and Crick proposed the double helix model, and they got Nobel Prize for their discovery. In 1953, 25th April, published one paper in Nature Journal, that molecular structure of nucleic acid is structure for deoxyribonucleic acid. So they first proposed the structure of nucleic acid, and they got Nobel Prize for this discovery. So what is the DNA structure? Why it is called double-stranded helix? Why this is called double-stranded helix? Because the helix is double-stranded. Two strands are connected each other by nitrogenous base. This four type base: adenine, guanine, thymine, and cytosine. Adenine, thymine, and guanine cytosines are bond each other, and this called double-stranded DNA structure. And the second point, that is most DNA double helix at right-handed. You have some idea about right-handed. What is right-handed? You look in this picture, that is right-handed. That is right-handed, and that is left-handed. It is the DNA structure. Many other forms of the DNA are present in nature. That is the bDNA, zDNA. zDNA is left-handed, but the most of the DNA of or bDNA is right-handed. So first character is DNA is helical structure. Second character is this is right-handed helical structure. And sugar phosphate backbone. It is made up of sugar phosphate backbone. We discuss this in the next slide. That is the beauty of sugar phosphate bond. Look in this. Why the name deoxyribose? That is ribose sugar. That is ribose sugar. But here in 2 prime, 1 prime, 2 prime, 3 prime, 4 prime, and that is 5 prime. In this 2 prime position, there is only H, no, not OH. So deoxy, 2 prime deoxyribose, 2 prime deoxyribose. This is deoxyribose sugar, deoxyribose nucleic acid, and that is 3 prime OH, and this OH bonds with another nucleotide there. So always DNA is 5 prime to 3 prime. Why? Because one DNA, the head portion of this DNA is 5 prime phosphate, and the last part of this DNA, that 3 prime OH. So this is 5 prime to 3 prime. 5 prime to 3 prime. Always 5 prime to 3 prime. Number 1 position, 1 prime position have nitrogenous base, 2 prime position is deoxy, 3 prime position is connected with other phosphate, 4 prime position attached with 5 prime position, and 5 prime position contain phosphate group, phosphoric acid group. This is always another character, the DNA having negative charge because this phosphoric acid group. Okay. And another significant character of DNA double helix, that this is antiparallel. At first I discuss about why parallel. Why parallel? Because always purine binds with pyrimidine. So 1 to 3 ring. Always purine binds with pyrimidine or pyrimidine. That is always 3 rings. Where purine binds with purine, that is 4 rings. So the parallelicity is destroyed. But in this case, always purine 2 ring binds with 1 ring. For this cause, for this reason, that is DNA is always parallel. But why anti-parallel? Because you look in this picture, because this is the 5 prime to 3 prime end of the on strand. This is 5 prime to, this is phosphate group, this is ribose sugar, and this is the nitrogenous base. But it binds with another strands with, that is nitrogenous base, that is pentose sugar, and that is phosphate group in opposite direction. That is, this is 5 prime to 3 prime, and the second group, that is 5 prime in the bottom, that is 5 prime, and in the top, that is 3 prime. So the DNA double helix is antiparallel, which means 5 prime end of the strand is paired with 3 prime ends of its complementary strand. 5 prime ends paired with 3 prime ends of this complementary strands, and vice versa. So this is antiparallel. We have some another forms of DNA, that is A form DNA, Z form DNA. I discussed in previous slide that P form DNA, that helical is right-handed, major group is present. What is major group? What is major group? This is major group, this is minor group, this is major group, this is minor group. Okay, so here major group is present, major group is present, major group, minor group, major group, minor group. This is right-handed. In A form, that is also right-handed, but in the Z form, that is left-handed. Major group in present in this in this DNA structure. Also, major group is present in A form of DNA structure, but absent in Z form of DNA structure. Base pair for helical term is 10.5. Is 10.5. That is the structure. Here, this is the one turn containing 3.4 nanometer length and having 10.5 base pair. Okay, that is 10 having 10.5 base pair per turn. So one base pair like nearly 0.34 nanometer, and that is the width. Width of this DNA is 2 nanometer. Width of this DNA is 2 nanometer. Here 10.5 nanometer, but in A form that is 11. That is 11 base pair per helical turn, and in Z form or turn that is 12 base pair per helical turn. Glycosyl bond conformation anti. Here also anti. Anti means purines binds with pyrimidine anti. Here also purines binds with pyrimidine anti, but in Z form maybe anti, maybe syn, because purine binds with purine, pyrimidine binds with pyrimidine. Okay. Helix per base pair. Helix rise per base pair. There is 3.4 angstrom. There is 2.6 angstrom, and that is 3.7 angstrom. Base tilt degree of tilting. This is 6 degree, 20 degree, and 7 degree. That is the structure of bDNA, aDNA, and zDNA. I query in the first slide that is there any another compartment where the DNA is located? Yes, that is mitochondria and chloroplast. That is circular DNA. That is circular mitochondria and chloroplast having circular DNA. What are the similarities between mitochondrial DNA and chloroplast DNA? Mitochondrial DNA and chloroplast DNA single circular chromosome having single and circular. Both DNA are double stranded but copying multiple copies. Okay. Moreover, both are randomly distributed to daughter cell like nuclear DNA. And both the DNA, the mitochondrial DNA and the chloroplast DNA are small in size, having quite few genes, and there are multiple copies in the cell.